Let's talk about the first set of reactions in cellular respiration. These are referred to as glycolysis. If you translate the word glycolysis, it means breaking down sugar. This is a set of reactions that occurs in the cytoplasm, and it occurs in the cytoplasm of all types of cells, all domains, prokaryotic cells, eukaryotic cells, bacteria, archaea, and eukarya. It's actually a series of 10 chemical reactions, and each of those reactions has its own enzyme that catalyzes the reaction. It's a very complex process. To summarize, the glucose that enters glycolysis is going to be oxidized until it becomes two 3-carbon molecules of pyruvate. 2 ATP are actually going to be used to begin this process of glycolysis. In other words, the cell has to invest a little bit of ATP in order to start the process. But along the way of those 10 reactions, 4 ATP molecules will be rebuilt. As bonds in glucose get broken down and energy is released, that energy will be captured and used to rebuild ATP. So the sum of that 4 ATP being built, 2 ATP being used to begin the process, means that glycolysis ultimately results in the gain of 2 ATP. And this ATP that is rebuilt during glycolysis is made during the process known as substrate level phosphorylation. As glycolysis proceeds and glucose is broken apart into pyruvate, an electron carrier molecule is going to gain the electrons that are released. So the glucose again is being oxidized, the electron carriers are being reduced. The electron carrier that the cell uses for glycolysis is called nicotinamide adenine dinucleotide. That's quite a mouthful, right? We shorthand the name of that molecule as NAD+. The NAD+ electron carrier is going to gain the electrons that are released from the glucose. Remember, when we watch these types of oxidation reduction reactions occurring, we should follow the proton. Remember that the thing that gets reduced also tends to gain a hydrogen atom. Well, if you look at the electron carrier, NAD+, it's actually going to become NADH as it gets reduced. It's going to gain electrons and it's going to gain hydrogen atoms. Now, on this slide, there's a drawing of glycolysis. There is nothing for you to write down in your notes about this drawing. I just wanted to show it to you because I want you to see how complex a process this is. This is not simply taking a glucose and splitting it into pyruvate. This is a step-by-step process that carefully modifies this glucose molecule in a way that allows, again, a little bit of ATP to be made and electrons to be harvested. So glucose goes in, but we then create molecules like glucose 6-phosphate and fructose 6-phosphate and fructose 1,6-bisphosphate and on and on and on until finally we're left with pyruvate. 1 6-carbon glucose goes in, 2 3-carbon pyruvate come out. In addition, again, we reduce electron carriers. NAD becomes NADH. And we produce a little bit of ATP through substrate level phosphorylation. So that's the take-home message about glycolysis: what's going in and what's coming out. This is another drawing of the process of glycolysis. I like this one because, again, it focuses on sort of the summary of the process: what's going in and what's coming out. Glucose, of course, goes in, and a little bit of ATP goes in. Remember, the cell has to invest a little bit of ATP to make this process occur. But then what comes out is pyruvate, a little more ATP, and we are reducing NAD plus to NADH. Now, glycolysis has to be regulated. Remember, we said cells are very thrifty, and a cell is not going to go through the process of glycolysis unless it needs to. So that means that it's not going to happen at a maximal rate at all times. It's only going to happen at its maximal rate, at its optimal rate, when a cell has a need for ATP. So when there's enough ATP in a cell, in other words, when there's an increased concentration of ATP in a cell, the cell will slow down glycolysis by inhibiting one of the enzymes. It's a key enzyme. It's the enzyme that catalyzes step 3, as a matter of fact, out of the 10 steps. This enzyme is called phosphofructokinase, and we shorthand that as PFK. It's a kinase enzyme. And remember, kinase enzymes phosphorylate things. The inhibition of phosphofructokinase that occurs when there's plenty of ATP around is referred to as a negative feedback type of inhibition. And what that terminology means is the fact that there's lots of ATP around, it's going to feed back. It's going to give information back to the cell so that the cell doesn't make unneeded, unnecessary ATP. So we refer to that as negative feedback. When there's enough product, ATP, we're going to have a negative impact on the enzyme. We're going to down-regulate the enzyme. We're going to have the enzyme work less optimally. We're going to inhibit the enzyme. Okay? So how does this work? Well, when there's plenty of ATP around, the ATP molecule itself actually binds to phosphofructokinase. It doesn't bind to the active site. It binds at another place on the PFK. And when the ATP binds, it causes the enzyme to change shape. Remember, anytime you stick a molecule onto a protein, you're going to cause that protein to refold slightly. And that's what happens with PFK. So this is actually also an example of allosteric inhibition, which we talked about when we discussed enzymes. The ATP is the inhibitor molecule. It's binding somewhere other than the active site, and it's causing an inhibition or a reduction in the activity of PFK. It turns out that negative feedback inhibition is actually a very common type of inhibition that cells use to regulate enzymes. And they're showing us that in this diagram. Here is a series of reactions. We're shown some kind of molecule called A is being transformed into a molecule called B with enzyme 1. And then B is being acted on by enzyme 2 to create C. And then C is being acted on by enzyme 3 to create D. So a series of reactions is going on, just like in glycolysis, with a series of enzymes being involved. Once there's enough D in the cell, the D molecule itself is going to have a negative effect on the activity of enzyme number 1. It's going to inhibit enzyme number 1 by feeding back by sending the information, hey, there's plenty of D around, to have a negative inhibitory effect on enzyme number 1. And that's the same negative inhibition that we see on phosphofructokinase. The next step in cellular respiration is this reaction called pyruvate processing. Now, in eukaryotes who have organelles in their cells, pyruvate is going to travel from the cytoplasm, remember that's where glycolysis occurs, across the membrane and into the matrix of the mitochondria. That's where pyruvate processing is going to occur in eukaryotic cells. Remember, pyruvate is a 3-carbon molecule, and it's going to get oxidized to a molecule called acetyl-CoA that has 2 carbons and to a molecule of carbon dioxide. This is actually a one-step reaction. No ATP is going to get made during pyruvate processing, but the electrons that are released from pyruvate will need to be temporarily held onto by an electron carrier, and during pyruvate processing the same electron carrier is used. NAD+ will be reduced to NADH. Now, the enzyme that performs pyruvate processing is actually an enormous molecule. It's referred to as an enzyme complex, and it is very tightly regulated. Again, the cell is not going to want pyruvate processing to occur unless it needs it to occur. Now, this complicated, enormous enzyme complex is called pyruvate dehydrogenase, or PDH. The other thing to know about pyruvate processing is that this enzyme, PDH, requires a coenzyme in order for it to catalyze the reaction in an optimal way. Remember, we said coenzymes are these small sort of accessory molecules that help enzymes perform at their optimum. So the coenzyme involved in pyruvate processing is called coenzyme A. And during the actual catalysis, the CoA will become part of the product molecule. That's why it's called acetyl-CoA. So the pyruvate molecule, which has 3 carbons, is going to be transformed into acetyl-CoA and a molecule of carbon dioxide. Pyruvate is being oxidized, so electrons are being released, and they will be captured by NAD+. It will be reduced to NADH, and we will have moved further along in cellular respiration. Processing is tightly regulated. In fact, pyruvate processing is referred to as the key regulatory control point of cellular respiration. That enzyme, pyruvate dehydrogenase, is actually going to experience control both in a negative feedback mechanism and a positive feedback mechanism. In other words, when the concentration of ATP in the cell is high, when there's plenty of it around, the enzyme will be slowed down in its activity. That's, again, negative feedback. But when the concentration of ATP drops, when it's lower than it needs to be, the enzyme will actually have its activity sped up. That is positive feedback. So ATP is feeding back information that will help determine how active that enzyme needs to be. But here we also see NADH feeding back and acetyl-CoA feeding back. So increased concentrations of any one of those, ATP, NADH, and or acetyl-CoA, will cause a phosphorylation of pyruvate dehydrogenase. So in this case, we don't see the ATP molecule itself binding to the enzyme. We have a phosphate group that is added to the enzyme. And that's going to change the shape of the enzyme, and it's going to decrease its activity. The third set of reactions in cellular respiration is called the Krebs cycle. This set of reactions is also going to occur in the matrix of the mitochondria in eukaryotic organisms. The acetyl-CoA molecule that was produced during pyruvate processing is going to first be joined together with a molecule called oxaloacetate. This is why we call Krebs a cycle. I'll show you a picture of this in a minute. But acetyl-CoA, as it goes into this cycle, is going to be joined together with a molecule coming out of the cycle, oxaloacetate. And the two things together will start the cycle all over again. The cycle is going to oxidize acetyl-CoA to carbon dioxide. Along the way, NAD+ is going to be reduced to NADH, and we're going to introduce a new electron acceptor molecule. This one is called flavin adenine dinucleotide, or FAD, and it's going to get reduced to FADH2. You'll notice that FAD doesn't have a positive charge on it, so when it gains electrons, it's going to become FADH2. Just like we saw in glycolysis, it's going to take a little bit of energy to get the Krebs cycle going, and that energy is going to come in the form of GTP. But just like we saw in glycolysis, that energy is going to get recouped by building a little bit of ATP through substrate level phosphorylation. This is a diagram of the Krebs cycle. We don't need to know all these details. What we really need to know is a summary of what's happening during this process, what's going in and what's coming out. So here's our acetyl-CoA molecule, the product of pyruvate processing. And it's going to enter this metabolic cycle along with a molecule that's coming out of the cycle called oxaloacetate. And those two things together are going to produce a molecule called citric acid. The citric acid will then pass through this cycle, being oxidized back to oxaloacetate and to carbon dioxide. Here's the carbon dioxide. So the oxaloacetate is just going to keep cycling around and around and around. And along the way, we're going to oxidize the acetyl-CoA to carbon dioxide. NAD+ is going to get reduced to NADH. In addition, FAD will be reduced to FADH2. And we're going to make a little bit of ATP through substrate level phosphorylation. The Krebs cycle, like the other processes that we've talked about so far, is regulated. The cell is only going to let the Krebs cycle proceed when it needs it to proceed. But unlike glycolysis and pyruvate processing, there is no one enzyme that's being regulated during the Krebs cycle. In fact, several of the enzymes involved in Krebs are going to be regulated. Just as we saw in pyruvate processing, the Krebs cycle is under both positive and negative feedback. That means when products are high, and that includes ATP and NADH, the cycle is going to slow down. That's the negative feedback. When products are low, ATP and NADH, the cycle will speed up. That's the positive feedback. It's a good place for us to stop. We'll see you in the next video.